Hey everyone, Dr. Mungli here. In this video, I will be explaining you all about heme catabolism. Now, what happens when heme is catabolized? The catabolism of heme, it will be going on in three tissues, and that is in the spleen, in the liver, and in the intestine. Now, from where we get this heme? The majority of the heme that is undergoing degradation, it is coming from hemoglobin which is there in red blood cells. Now, how much hemoglobin is undergoing degradation in our body? Per hour, there will be destruction of 10 million red blood cells per hour. Remember, 1 hour. In 1 hour, 10 million red blood cells will be dying in our body. And around 6 grams of hemoglobin, it will be undergoing turnover per day in our body. And each gram of hemoglobin, it will be giving you a 35 milligram of bilirubin, which is a heme degradation product. Now, 1 gram of hemoglobin will produce 35 milligrams of bilirubin. If you do that calculation, 6 grams of hemoglobin, how much you are going to get bilirubin molecules when the 6 grams of hemoglobin is undergoing degradation. Now, overall, so heme degradation is giving bilirubin. So, the heme is coming from red blood cell which contains hemoglobin. That is 70 to 80% of the bilirubin that is produced in our body. It is all coming from hemoglobin by the destruction of the red blood cells. Now, the rest of the bilirubin, it will come from other heme containing proteins like cytochrome P450s, which is there in hepatocytes in the liver, and some other heme containing enzymes. It can be catalase, tryptophan, pyrrolase. So this kind of enzyme, the cytochromes present in the electron transport chain, these are heme containing proteins, and when these proteins undergo turnover, heme is converted into bilirubin. Now, in this video, I will be focusing more on how red blood cells will undergo destruction, what will happen to the hemoglobin, and what will happen to the heme, how exactly bilirubin is made, and what happens to that bilirubin, how we are going to excrete it out. So, the first tissue that is participating in the metabolism in the catabolism of heme is spleen. So, the reticuloendothelial system in the spleen, so it is the one which will take care of senescent red blood cells, so that mean to undergo destruction. So, reticuloendothelial system trap that senescent red blood cell, breaks open the red blood cell. So, extravascular hemolysis going on here, and hemoglobin comes out of red blood cell, and from hemoglobin, so HB, that is hemoglobin here, so heme is separated from hemoglobin, and globin is separated out. Now, what happens to the globin? So, globin will go into amino acids pool. So, whereas heme that is there here, so this heme, it will get into heme oxygenase reaction. So, the heme oxygenase is present in the spleen, the cells present in the spleen. So, what will happen to the heme here? So, heme oxygenase, it is going to break a tetrapyrrole ring, so giving rise to biliverdin. So, there will be formation of biliverdin. So, this particular job, it will be done by an enzyme called heme oxygenase. So, what this heme oxygenase does, it is going to break the tetrapyrrole ring. You know the tetrapyrrole ring as it is shown in the figure there. So, it has got ferrous iron in the center. That's the heme with tetrapyrrole ring which are held together, 4 pyrrole rings held together by methynyl bridges here. So, heme oxygenase, what it does, it is going to open the tetrapyrrole ring and makes a biliverdin molecule. During this process, the ferrous iron which is there in the center of the heme, it will be released as ferric iron. So, ferrous iron is taken out of this molecule and released as ferric iron, and this ferric iron will be stored as ferritin. So, that's the fate of ferrous iron present in the heme, and that will undergo a recirculation process. And also during this reaction, there will be release of carbon monoxide. So, there will be release of CO, carbon monoxide. And in fact, this is the only source in our body. So, heme oxygenase catalyzed reaction where heme is broken down into biliverdin is the only reaction in our body which will give carbon monoxide. So this carbon monoxide can be exhaled out. So we can measure breath carbon monoxide levels, and the breath carbon monoxide levels is an indirect indication that the amount of hemolysis that is going on in our body. So higher the hemolysis, more the carbon dioxide in the breath. So that's one of the applied aspect here you got to remember. So the carbon monoxide, if you measure in the breath, so that is an indirect indication that how much hemolysis is going on, because this is the only reaction in our body which makes carbon monoxide here. That's an application. So the once heme is converted into biliverdin. Biliverdin is in fact it's a green pigment. This is green in color. And this biliverdin, it will undergo further reaction by biliverdin reductase enzyme. So the biliverdin reductase converts biliverdin into bilirubin. So the enzyme is biliverdin reductase. So during this reaction, what will happen? So there will be reduction in the methanol bridge between the third and the fourth pyrrole ring into methylene bridge. So the methanol bridge is reduced into methylene bridge, especially between third and the fourth pyrrole ring. And for that, you need NADPH molecule. So NADPH is needed to do this function, NADPH plus H plus, and also for heme oxygenase you need NADPH plus H plus. So you all know NADPH plus H plus, it will come from pentose phosphate pathway. So this is how biliverdin is converted into bilirubin, and bilirubin is yellow in color. This is an yellowish molecule, whereas biliverdin is green in color. And also note that the type of bilirubin that is synthesized in the spleen predominantly, so it is an unconjugated bilirubin, unconjugated, and it is hydrophobic. So water insoluble bilirubin, unconjugated, is not conjugated with anything. Solubility of this bilirubin is very low. It is a lipid soluble molecule, but it's hydrophobic. It is not soluble in the water. So this is an excretory product here, which is a degradation product of heme, is the bilirubin. Right now it is water insoluble. So in order to excrete this out, this is an excretory product. It has to be thrown out of our body. So in order to excrete this out, so bilirubin has to be sent to the liver from the spleen. So we are in the spleen right now. So all these reactions are going on in the reticuloendothelial system, especially in the endoplasmic reticulum of reticuloendothelial system in the spleen. So you got bilirubin here, and this bilirubin has to be excreted. So it has to be sent to the liver to make it water soluble, because the molecule that need to be excreted, they need to be in water soluble form. So we need to convert that unconjugated bilirubin, which is hydrophobic, into conjugated bilirubin, which will be hydrophilic. So that job, it will be done in the liver. Let's see what happens in the liver, how to take this unconjugated bilirubin from the spleen into the liver to make it conjugated. Alright, now let's see what happens to unconjugated bilirubin in the liver. So let's write liver now. So we will be in liver. So all the reaction that I'll be explaining, so they will be going on in the liver now. So what we do is, so bilirubin, which is made in the spleen, it will be secreted into the blood. So that it gets into the blood, bilirubin comes into the blood. So I'll write it as unconjugated bilirubin, UCB, unconjugated bilirubin, UCB. So this unconjugated bilirubin, it will be binding with albumin in the blood. So it binds with albumin because albumin is keeping it in soluble form. So unconjugated bilirubin binding with albumin, so it will be taken to the liver. So that is the transporter of unconjugated bilirubin in the blood. And also note that albumin, it has got two binding sites for unconjugated bilirubin. One is a high affinity binding site, which has got a capacity to bind up to 25 milligrams of unconjugated bilirubin per 100 ml of blood. Anything in excess of 25 milligram per deciliter of the blood, unconjugated bilirubin, it will bind with the lower affinity binding site. So the low affinity binding site. So unconjugated bilirubin binding to the high affinity binding site, so it is not easily diffusible form of bilirubin there, whereas the unconjugated bilirubin that is binding to the lower affinity binding site, it can be diffused into the tissue. That is why whenever unconjugated bilirubin levels in the blood exceeds 25 milligram per deciliter, it can diffuse into the tissues and it can damage the neuronal tissue. And one of the conditions where you can see the neuronal damage because of elevated levels of unconjugated bilirubin more than 25 milligram per deciliter. So that the disorder is Kernicterus. So in Kernicterus, so there is elevation of bilirubin more than 25 milligram per deciliter, which will bind to the lower affinity site and then causes diffuse into the neuronal tissue and causing neurological condition called Kernicterus. So which is commonly seen in premature babies where there is a decreased conjugation process. Anyway, so when albumin binds to unconjugated bilirubin, and also under normal conditions we have unconjugated bilirubin which is around 0.8 milligrams per deciliter, because normal total bilirubin is 0.3 to 1.3 milligram per deciliter. In that 80% is unconjugated, 20% is conjugated. So if you go by that calculation, so unconjugated bilirubin will be around 0.8 milligrams, which is quite sufficient. Albumin has got capacity up to 25 milligrams. So it is normally we have 0.8. So it generally doesn't bind to low affinity binding site, it will be binding to higher affinity binding site. Anyway, so once albumin is carrying it to the liver, so what we will do now is I am going to take this albumin to the liver, albumin bound unconjugated bilirubin, so to the liver. So in the liver what happens, so means over the surface of hepatocyte the albumin is separated and unconjugated bilirubin gets into the liver. How unconjugated bilirubin will get into the liver? So the unconjugated bilirubin is taken into the liver by a bilirubin transporter. There is a transporter over the hepatocyte called bilirubin transporter. So this bilirubin transporter, it is a high capacity transporter. It has got a large capacity to take up as much unconjugated bilirubin into the tissue. And also note that bilirubin transporter is a bidirectional transporter. It means if there is more bilirubin inside the hepatocyte, it can be thrown out into the blood, or if there is more bilirubin in the blood, it can go into hepatocyte. Now let's see what will happen to unconjugated bilirubin in the liver. So liver, the role of liver here is to conjugate this hydrophobic unconjugated bilirubin into hydrophilic conjugated bilirubin. So the main role of liver is to conjugate bilirubin so that it can be excreted out of our body. So how to do that? So this unconjugated bilirubin, before it undergoes conjugation process, so it transiently it will react with glutathione S transferase GST enzyme, and later GST is released and it will undergo conjugation process. Let's see what is the conjugating process here. So the unconjugated bilirubin, that is UCB, is converted to bilirubin monoglucuronide, which is basically a conjugated bilirubin there. So the process here is it will be added with UDP glucuronate. UDP glucuronate gets into the reaction, UDP goes out and glucuronate will bind. So that is why it is called as bilirubin monoglucuronate. So the job is done by an enzyme called UDP glucuranosyl transferase 1A1, which is simply written as UGT1A1. This is the name of the enzyme. I will repeat again. The name of the enzyme is UDP glucuranosyl, that is UDP glucuranosyl transferase. It is going to transfer this glucuronate, which is coming in the form of UDP, to unconjugated bilirubin and make it bilirubin monoglucuronide. That's why the name of the enzyme is UDP glucuranosyl transferase 1A1. 1A1 is a isoenzyme here. So it is responsible for conjugating unconjugated bilirubin into conjugated bilirubin. Now, furthermore, bilirubin monoglucuronide will undergo similar reaction where one more UDP glucuronate get into the reaction, UDP goes out and glucuronate will bind. So you get bilirubin diglucuronide, and that is also conjugated bilirubin. So basically you are conjugating unconjugated bilirubin into conjugated bilirubin by adding two glucuronate molecules. One glucuronate, bilirubin monoglucuronide, second molecule of glucuronate, bilirubin diglucuronide, and the job is again done by the same enzyme, UDP glucuronoside transferase 1A1 enzyme. Okay, now this is the conjugated bilirubin, which is water soluble bilirubin. Now, any defect in these enzymes, say defect in UDP glucuronoside 1A1 enzyme, it can give rise to a disease. If there is complete absence of UGT1A1, this will give rise to a disorder called Crigler-Najjar syndrome type 1. And if there is a deficiency of UGT1A1 enzyme, it will give rise to Crigler-Najjar syndrome type 2. So, complete absence of UGT1A1 is Crigler-Najjar syndrome type 1, and partial deficiency is giving rise to Crigler-Najjar syndrome type 2. Because of this enzyme deficiencies, there will be elevation of predominantly unconjugated bilirubin in the blood, giving rise to signs and symptoms. So, I'll come to those disorders later, so probably I will make a separate video on those disorders. Now, what will happen to this conjugated bilirubin here? Once you are conjugating this bilirubin, so this is now ready, it is a water soluble bilirubin. This is a conjugated bilirubin, water soluble. So, this is ready for excretion now. So, how this molecule will be excreted? Where it will go? So, the conjugated bilirubin, it will be secreted out of the liver into biliary canaliculi. It gets into biliary canaliculi, and that job it will be done by a secretory protein which is there over the hepatocyte, and the name of that protein is MOAT. Name of the protein which is helping in the transport of bilirubin monoglucuronide and bilirubin diglucuronide. Even monoglucuronide also can undergo transport process like this. So, predominantly bilirubin diglucuronide. So, the multispecific organic anion transporter, that's what is MOAT. Multispecific organic anion transporter is going to secrete, it's an active transport process where ATPs are spent. It's a ABC type of transporter, ATP binding cassette transporter. BDG is secreted into biliary canaliculi, and from there it will be getting into gallbladder. It gets into the gallbladder, and it will be stored until gallbladder is contracted. So that's what happens to your conjugated bilirubin, which is made in the liver. Ultimately, it ends up in the gallbladder. So what will happen when the gallbladder contracts? Alright, now let's see what happens to this bilirubin which is getting into the gallbladder. So the conjugated bilirubin which is getting into the biliary canaliculi and then accumulated in the gallbladder. So when the gallbladder contracts in the presence of cholecystokinin, so the conjugated bilirubin gets into the duodenum. So from the duodenum, so it is getting into jejunum and it's not absorbed. Conjugated bilirubin is not absorbed. It going to the ileum. So especially the terminal part of ileum and in the colon. So this is where the intestine comes in now. The third organ where there is a metabolism of bilirubin is going on, it is in the intestine. So especially terminal part of ileum and the colon. So the conjugated bilirubin that is present here coming from the gallbladder, so it is converted into unconjugated bilirubin. So this job it will be done by bacterial beta-glucuronidases. So these are bacterial enzymes. So bacterial beta-glucuronidase. Bacterial beta-glucuronidase enzyme converts conjugated bilirubin into unconjugated bilirubin by releasing glucuronate. Glucuronate is released. So that's unconjugated here. So furthermore, unconjugated bilirubin is converted into urobilinogen. So urobilinogen is made and that is a bacterial oxidation. Again, this is an oxidation process. So the bacterial oxidation converts UCB, unconjugated bilirubin, into urobilinogen. So what will happen to urobilinogen? 80 to 90% of this urobilinogen, it is excreted in the stool. So it gets into the stool. So urobilinogen 80 to 90% gets out of our body in the stool. That's how we are excreting this bilirubin. So heme converted to biliverdin, biliverdin into bilirubin, which is conjugated in the liver. So becoming conjugated bilirubin, getting into the intestine, converted to unconjugated bilirubin, further converted to urobilinogen, and that urobilinogen gets into the stool, 80 to 90% of it. So urobilinogen that is present in the stool, we also sometimes refer it as stercobilinogen. So stercobilinogen can be oxidized into stercobilin, and that will give the normal yellow color of the stool. Okay. Now 10 to 20% of this urobilinogen, it will get into enterohepatic circulation. 10 to 20% enters into enterohepatic circulation. So it will be going into the liver. It will be taken up by the liver. So liver is going to take that. So that's 10 to 20%. In that 10 to 20%, some amount of that urobilinogen, so around less than 3 milligrams of urobilinogen, it enters into systemic circulation and it will get into urine. It will be filtered by the kidney and into urine. So you do see some amount of urobilinogen in the urine, and that is the one which is responsible for normal yellowish color of the urine, because some amount of urobilinogen which is getting into enterohepatic circulation, so it escapes that enterohepatic circulation, gets into the systemic circulation around less than 3 milligram of it, and it is eventually filtered by the glomerulus going into urine. So that's the normal color of the urine. So this is what is all about metabolism of bilirubin, that is hemoglobin breaking down into heme and globin, and heme converted to bilirubin, going into the liver, is conjugated, and then coming into the intestine, and in the intestine it is unconjugated, further oxidized into urobilinogen. Most of it is excreted in the feces, some of it is excreted in the urine. So this is all about catabolism of heme and formation of the bilirubin and metabolism of the bilirubin metabolism, how it is conjugated, how it is excreted and all that. So I hope this video has helped you in understanding heme catabolism process or heme degradation process. If you have any questions, put that question in the description, in the comment section below. And also, if you like the video, give thumbs up, and for more videos like this, you can consider subscribing to this channel so that you get regular updates. Thanks for watching.